We begin the second video lecture with a very odd request. Take two fingers and hit yourself smartly on the inside of your forearm. Not too hard, but not too soft. Describe what you see. You should see a red area. Obviously, you're going to feel a little pain. And if you observe closely, you'll maybe feel a little heat and a little swelling. This process is one of the most important defenses your body has. It's called inflammation. On the cellular level, inflammation is caused by damage, either physical, as you just did, or by infection. When damage is detected, your cells will release signals that begin the process of inflammation. This results in vasodilation and increase in vascular permeability. Fluid proteins and cells then flow into the area. The target tissue becomes red from red blood cells, heated from the release of what are called pyrogens. It swells due to the fluids that come in, and it becomes painful. Again, inflammation is the most important defense that your body has. Its initial response to any pathogen, and what it does is it focuses the immune system on the actual problem in your body. Now, inappropriate inflammation can cause illness, especially in people as they age, and this is an active area of research. Triggers of inflammation are damage to the host, certain immunological reactions, and then, of course, harmful agents such as pathogens. Cardinal signs of inflammation are swelling, again, caused by gaps between capillary cells, redness, caused by the flow of blood into the area, heat, which is the accumulation of blood in the area, and also the release of pyrogens, and then pain. And this is a response to tissue damage. It's an irritation of sensory nerves in the affected area. The things that signal inflammation are danger-associated molecular patterns and microbe-associated molecular patterns. And as you can see, this graphic goes through the process. In the first step, the skin is broken, allowing access to the pathogens and releasing PAMPs and DAMPs. Mast cells detect the PAMPs and DAMPs and release the meteors of inflammation. The mediators of inflammation, not the meteors. I'm going to leave that. Moles raise the alarm and cause blood vessels to open in the area, allowing access to phagocytes. Phagocytes then attempt to neutralize the pathogens and clean up any damage. Platelets also enter the area and begin the repair process. And finally, the influx of blood and fluid causes the swelling, pain, and heat associated with inflammation. In some cases, this will take care of an infection. A lot of the time when you get cuts, it doesn't turn into an abscess because your body deals with it. Even if you get a viral pathogen that starts infecting your skin, the inflammatory process on its own may take care of it. Now, one of the things that we mentioned in the inflammatory process was the presence of phagocytes. So let's take a deeper dive into what those are. Phagocytes differentiate in the bone marrow. They originate there, and under the influence of cytokines become polymorphonuclear leukocytes or monocytes. The major types of phagocytes that I'm going to talk about are neutrophils and monocytes and macrophages, and they make up a significant majority of the phagocytes in the body. There are others, but we're not going to talk about them here. Neutrophils, as I said, are also called polymorphonuclear leukocytes. They're one class of polymorphonuclear leukocyte. They're the most common. They make up 90% of the granulocytes in the body. They leave the bone marrow and circulate in the bloodstream. They have a very short lifespan, dying after only a few days. They are normally the first phagocytic cells that encounter an infection, and they can rise to large numbers during a severe illness to help you fight that infection. Your bone marrow contains a large reserve of neutrophils that are stored there and are released when needing to fight an infection. The neutrophils travel to enter the infected tissues where they engulf and kill bacteria or whatever is causing the problem. The neutrophils die in the tissue and are engulfed and degraded by macrophages. Monocytes and macrophages are the other type of phagocyte that we're going to talk about. Monocytes differentiate in the bone marrow, and they have an unsegmented nucleus, which differentiates them from the neutrophils. They are much longer lived. After they are produced, they circulate in the blood for a period, then settle into a tissue and mature into a macrophage. Macrophages are mature monocytes that are attached to lymph tissues. They can be up to 10 times the size of a monocyte, and these phagocytes contain lysosomes, as all of them do. When the phagocyte attacks, it will annihilate whatever it engulfs. All right, so the roles of macrophages. They remove our own dead cells when they reach the end of their useful life. They remove pathogens. They are also important in the creation of immune proteins and peptides. Things like transferrin, which is an iron-binding protein. Complement proteins are synthesized in macrophages, and certain cytokines are produced by them. Now, both neutrophils, monocytes, and macrophages can go through the process of phagocytosis. So let's spend some time talking about that. The first part of phagocytosis is detection, and the detection of pathogens involves sensing their presence, and this can be done in a number of ways. First of all, phagocytes have receptors that will recognize N-formyl methionine in proteins. Only bacteria form proteins with an N-formyl methionine on them, and if a phagocyte detects that, they will start to migrate towards that area. Remember that complement releases C3A and C5A. These are chemoattractants for phagocytes. Finally, the mediators of inflammation, these compounds that get released, such as histamine, will attract phagocytes to an area. Okay, so the phagocyte gets into an area and detects undesirable organisms, let's say a bacterium. First of all, they will attach to it. Now, this is a very inefficient task without help, and at the worst case scenario, they can trap the target bacterium against the wall and then go envelop around it. This is non-specific and it's slower. However, phagocytes have 40 receptors on their membrane surface to help. These receptors recognize molecules that enhance the binding of phagocytes. And this process, this enhanced binding, is called opsonization. Macromolecules that do this are called opsonins, and they speed phagocytosis. And what's happening is these opsonins provide a molecular handle that the phagocyte can grab onto. Two examples are IgG antibodies and the complement protein C3b. Remember, when we talked about the complement cascade, the C3b protein is lodged into the membrane during the complement cascade of the target bacterium. And then now the phagocyte can use that to grab it and engulf it. The phagocyte is now attached to the bacterial target, and it's time for ingestions. Attachment causes a signal that triggers ingestion. The target is encircled in a membrane called a phagosome. And this is somewhat similar to endocytosis, except you're bringing in a bacterium, not some molecule that you want to transport in the cell. This process requires ATP, and the phagocyte will shift to a fermentative metabolism. It does this to generate a lot of energy and to produce acidic products that it uses to attack the bacterium with. The phagosome will then merge with a lysosome, and the lysosome is full of toxic chemicals and enzymes that will kill the bacterium. The lysosome, when it fuses with the phagosome, will explosively discharge its toxic substances at the bacterium. So how does the phagolysosome kill? The phagosome is a deadly environment for bacteria. Numerous mechanisms eliminate bacteria once they are inside, and these can be divided into oxygen-dependent mechanisms and oxygen-independent mechanisms. Oxygen-dependent mechanisms are the most effective. Two examples are the creation of hydroxide radicals and the creation right here. You see this? Oxygen comes in, hydroxide and superoxide is made, and then you get hydroxide radicals, right? And the creation of hypochlorous acid by myeloperoxidase. In this case, the superoxide dismutase is turned into hydrogen peroxide. This is combined with chlorine to make hypochlorous acid, which is very deadly. And both of these compounds will react with bacterial DNA, proteins, and other cellular constituents, rapidly inactivating them. The other part of this process is oxygen-independent mechanisms. And this includes a collection of enzymes such as lysozyme, glycolases, phospholipases that degrade membranes, nucleases, lactoferrin that steals iron away from them, and proteases. All of these combine to make it much more difficult to grow. Most bacteria will only survive for a few minutes once they enter a phagolysosome. So this is a very effective way of killing them. Once you're done with killing the microorganisms, you have egestion and antigen presentation. Now, neutrophils don't do this. They just accumulate junk inside phagolysosomes. They fill up with phagolysosomes and then they die. Macrophages and monocytes live much longer. They will expel unwanted material by reversal of phagocytosis, which is called egestion. And some parts of this material are saved to make antigens to present to the rest of the immune system. Okay, that's what phagocytes do. I want to mention a few other things before we finish up innate immunity. This brings up natural killer cells, which don't really fit well in either category of innate immunity or acquired immunity. This illustrates that the immune system evolved over millions of years and has various components that don't fit nicely into our categorizations. Sometimes that's how it is, and frankly, biology just doesn't care as long as it works. These are non-phagocytic granular lymphocytes present in the body at low populations. They have a small amount of immunoglobulin and MHC1 molecules on their surface. Again, we'll get back to these later. And they're not T cells, but their behavior is similar to that of a cytotoxic T cell. We'll be talking about those in later lectures. They seem like they are not inducible. They also have Fc receptors on their surface that are capable of binding IgG, and this will trigger attack by the natural killer cell. It's definitely not adaptive immunity, but it has some of the features of T cells. So I just want to make you aware of what these are. Another thing that you'll see that's at the interface of the innate and adaptive immunity is dendritic cells. They are part of the innate response, but they also initiate adaptive responses such as antigen presentation. They will produce cytokines, and they also recruit leukocytes. We'll have more to say about them when we talk about acquired immunity. There are many cells involved in the immune response, and it's important for them to communicate. This communication occurs using cytokines, which are small peptide hormones secreted by cells. In the case of the immune system, cytokines will signal the presence of an invader and induce an immune response, will turn on and off appropriate immune cells, and will shut down the immune response at the end of an infection. Shown here is an example of the response of a macrophage upon sensing microbial products. It will release 5 different cytokines: interleukin 6, tumor necrosis factor alpha, interleukin 1 beta, interleukin 8, and interleukin 12. Each of these has various effects on the body as shown, and that will then marshal the rest of the immune response. And no, I don't want you to memorize these effects. It's just for illustration. I do want you to understand generally what cytokines are doing. Here are some more examples of what cytokines do in the body. All right, so interleukin 1, it comes from macrophages, and it activates inflammation, fever, and activation of the liver, right? Interferon gamma is secreted by natural killer cells and by T cells, and it activates macrophages, etc. So you can see each one of these signals communicates to another part of the immune system and then has an effect. I want to take a second. I want you to do a 1-minute paper. And in it, I want you to compare macrophages to neutrophils. What do they have in common and how are they different? What this means is take about a minute and write down everything you can think of about these two things. Summary of the innate response. The innate response consists of physical barriers, chemical barriers, intraepithelial lymphocytes, and antimicrobials that will attack bacteria. Phagocytes, such as neutrophils, which are first responders, and monocytes and macrophages, which are recruited and are the real start of the innate response. 3, there's an increase in numbers of these immune cells, which is stimulated by cytokines. 4, neutrophils migrate to extravascular sites by binding to endothelial adhesion molecules and squeezing through and getting into the area. Microbes are then recognized and destroyed. How they are destroyed and how successful they are depends upon how many microorganisms and how virulent the pathogen is. Is innate immunity enough? The innate immune response will attempt to contain and destroy the infection. Now, microbes grow rapidly, and some of them have figured out ways to outsmart the immune system. But the immune system is situated all over the body. As you can see here, this diagram shows you all the lymph vessels all over the body on one side of the body. And again, some student asked me this question, is the lymph system only on the right side? The answer is no. It was just drawn on the right side. These vessels are situated all over the body, and they monitor what's going on. When does the adaptive immune system kick in? When the innate immune system is overwhelmed. In some cases, the ubiquitous responses of the innate immune system that happen in 0 to 4 hours can stop and remove the infectious agent. In some cases, the induced responses of innate immunity, such as phagocytes, etc., are activated. They then deal with the effector and remove the infectious agent. If these two fail, then the adaptive response comes forward with B and T cells and tries to remove the effective agent. And that is the topic of the next lecture.